it's such a beautiful day. We decided to head out, pick a few flowers, and do an investigation using flowers. So, oh, we've got a few there. Can you get those?
Yeah, I'll get those.
So we're back indoors with plenty of colorful flowers. What have we got here?
So we've got a periwinkle, we've got a rose, and we've got some other flowers here.
So what we are going to be doing is we're going to look at the flower externally and then have a look at the pollen grains under the microscope. And you can design different investigations around flowers. Which type of flower would you find in a particular area? What would the arrangements of the stamen be? You could look at the different pollen morphology. Many things can be done, and we are going to get ours started.
Yeah.
Begin with the external morphology first. So as we see here, we removed the petals, then the sepals, and reached the stamen and the pistil of the flower. The stamen is the male reproductive part. As you can see here, I'm pointing towards the anther and the filament. The little stick is the filament, and the little round blob on top of it is the anther, and that's where we find the pollen grains. Some flowers have only female reproductive parts, some flowers have just male reproductive parts, but I chose to go with the shoe flower or the hibiscus because that gives a beautiful representation of a typical flower with both the male and the female parts. So now zooming in, we are going to slowly remove all the anthers and the filament, get rid of the stamen, and then reach the female reproductive part of the flower. So for my investigation, I decided to look at the stamen morphology and the pollen morphology of flowers which are pollinated by bees and represent my findings using a poster. And I also decided that I'll be looking at tropical flowers which are found in Australia and not just general flowers. So that sort of allows me to get more specific with my findings. And here you can see I've arranged the pistil or the female part of the flower. And it's got three parts: the stigma, which is on top, it's sticky for the pollen grains to stick. Then we've got the style and the ovary where the egg cells are found, and those eventually end up becoming seeds. So the pollen grains get stuck to the stigma, and the pollen tube then travels and sends the male cell into the ovary, and that's where fertilization takes place and the cycle continues. We decided to make a little illustration of the different parts of the flower with the stamen and the pistil. And now we move on to looking at the pollen grains. So here we are making a little wet mound, taken a slide, a drop of water, and that is the anther of the flower. Now as you can see, if we zoom in, those little white grains, that's actually the pollen. Now we'll put the cover slip, and you can use any microscope. This is the one that we got, I think, from Kmart. It's pretty neat. It doesn't really have to be very sophisticated. Just remove the excess water and make sure the cover slip is not dancing. That'll sort of hold it in place, and then go for it. Look at it. So we decided to look at it under 40x. Here you can see the hibiscus pollen, and this is rose. And we did it with all the other flowers as well, and eventually I documented everything in this e-poster. And my question was, which stamen arrangement and pollen grain structures are most commonly seen in tropical angiosperms undergoing melitophily, which basically means pollination using bees. And here you've got the introduction, the methodology, results, discussion, and conclusion, and also things that could have been done better. For example, maybe I could have collected a bit more flowers and probably had bigger pictures. So there's always scope for improvement, isn't it? So have fun.